It began with a single question: what happens when you combine natural harmony, scientific precision, and ambition? We didn't just want to build projects, we set out to prove what was possible.
Over the past year, the velocity hasn't stopped. We've expanded our initiatives, welcomed brilliant minds to the collective, and pushed the boundaries of our core ventures. Every test, every breakthrough, every sleepless night was a stepping stone toward something far bigger. Today, that foundation brings us to now. As Sri Lanka's first synthetic biology and genetic engineering venture, we engineer real-world solutions across agriculture, the environment, and human health, backed by precision molecular services and specialized R&D through Bioloom Islands, Think Labs, and Scientifics. We prototype biological hardware, develop advanced research kits, and consult with industry leaders to turn theory into viable technology, and through our internship foundry.
The Bioloom Islands internship is a guided, immersive experience that bridges academic learning with real-world application. Develop research ideas, build experimental frameworks, and gain hands-on laboratory experience. Work on live commercial research projects and learn from global mentors. Choose your pathway, build your future, and join us at Bioloom Islands.
Welcome to Bioloom Islands. Bioloom Islands is a learning space built for the next generation of biologists. A place where molecular biology, computation, and curiosity intersect. Biology is evolving faster than traditional education can adapt. We introduce the new era of human and environmental evolution: synthetic biology. Yet many learners never get the chance to use them meaningfully. Bioloom focuses on practical understanding. We turn complex tools into usable skills through hands-on workshops, guided exploration, and real scientific workflows. Today's workshop is one island in that larger journey.
All right, good evening, everyone, and a warm welcome to all of you joining us tonight from across Sri Lanka. And I can see from, and I will be your host for next hour. So, Engineering Biology with Foundational Models, a workshop that Bioloom Islands will be running on the 5th of September. Just a reminder, tonight is just the trailer, not the film. So within the next 60 minutes, we want to do three things for you. Show you what has actually changed in biology over the last 2 years, and let you hear it from the people who work with those tools every day, and walk you through exactly what the workshop covers so you can decide whether it is for you or not. Before we get started, just a few quick housekeeping notes. First, everyone will be muted upon entry. That's intentional, as it helps us to keep the audio clear and ensure everyone can hear comfortably. Second, please feel free to use the Q&A box or the chat throughout the session. Don't wait until the end to ask your questions. Simply drop them in as they come to you, and I'll collect and address them along the way. And finally, yes, the session is being recorded. We'll be sharing the recording and the slides with everyone who's registered, so you'll have the access to them after the session as well. And one more thing, which I will mention now and come back to properly later as well, because I do not want anybody to miss it. At the end of the tonight's session, we are putting a promo code on the screen. The workshop is priced at 5,000 rupees. With that code, it becomes 3,500, and it is only for the people who are here tonight in the room. So please stay until the end. The code goes up live, and I will hold it on the screen for long enough for everyone to write it down. So with that, let me tell you who's behind tonight's session. Bioloom Islands is Sri Lanka's first synthetic biology company. I want to sit it on that sentence for a second because it's easy to say quickly and easy to underestimate. Synthetic biology is the discipline that treats living system as something you can engineer. You design a genetic sequence the way an engineer designs a circuit, you build it, you test it in a cell, and you learn from what comes back. That discipline has built a multi-billion-dollar industry in the United States, in the United Kingdom, in Singapore, and in China. Until Bioloom, it did not have a company of its own on this island, and it does now. Our research and development runs across three core divisions. The environmental division works on Sri Lanka's environmental problems, from coastal pollution to inland water contamination. The health division goes from engineering novel therapeutic pathways through the commercializing diagnostic-led preventive care. And the agriculture division takes on coastal soil salinity, crop pests, and post-harvest loss, which in this country is not a small number. Around those three sit our specialized division, Science Outreach and Communication, the Academic Division, and Global Partnerships, which is how we stay connected to biotech leaders outside the island. On the services side, there are 6 ways we work with people: consultation, research planning and experimental design for researchers, institutes, and businesses, research-focused internship that puts students on real projects at the bench and on the computer, tailored workshops, and terrain built for a specific university or organization. And then there is the third pillar, which is why we are all here tonight: education. Bioloom Island Knowledge is Sri Lanka's first dedicated biotechnology learning platform. Synthetic biology, CRISPR and genome editing, cell biology, biopharmaceuticals, bioinformatics, agricultural biotech, metabolic engineering, biosafety, and ethics. Built for people in this country, we think of those 3: research, services, education as one thing, not 3. You can build an industry with laboratories and no people, and you cannot build it with trained people and know where for them to work. Tonight is us working on the people half. So why this topic and why this year? For most of the history of molecular biology, our bottleneck was data. We could not read genomics fast enough or cheaply enough. That bottleneck is gone. The first human genome took 13 years and something close to $3 billion. Today, a human genome is sequenced for a couple of $100 in about a day. Lab automation, liquid handlers, robotic colony pickers, Claude Labs is collapsing the build and test house. AI foundation models are collapsing the design and the learn halves. When both halves collapse together, a loop that used to run for times a year starts running 4 times a month. And the lab that runs its loop 10 times faster than yours is not 10% better than you. Within a couple of years, it is in a different conversation altogether. That is the thing I want you to hear clear tonight, and I would rather say it plainly than diplomatically. Nobody in this room is going to be replaced by an AI. But the researcher who has these tools in their hands is going to move very quickly past the researchers who does not. In publications, in grant applications, in industry roles, and in what they are able to even attempt. That gap is opening now, and it is far easier to step across it now than it will be in 3 years.
happening is that Anthropic is calling this an AI workbench for scientists, and this platform connects to tools like research databases, and it actually analyzes data with specialties like genomics and structural biology. And the idea here is to use AI to accelerate drug development, so making drugs better and making them faster. And Anthropic was rolling this out, like I said, this event today in San Francisco, and they had executives announcing that Anthropic is also going to start its own drug discovery program. And the executives were careful to position this as a way for Anthropic to make sure that it's building the best models, tools, and products for the drug makers that it's trying to sell the Claude science product to. And they said that Anthropic will focus on neglected diseases that traditional biopharma companies wouldn't find attractive, and that this will be an early stage preclinical work. And what happens if they actually come up with a promising drug? Well, we asked Anthropic, and they said that they're at the early start of this and that they will share more as the work progresses. But that's the question, right? And we've seen plenty of other tech companies try to get into healthcare. You have Alphabet, Amazon, Apple, all those come to mind. So it's not unusual, but this certainly could be an interesting one to watch.
Yes, John. So basically, just like we have AI predict our language in sentences with syntax in English or other languages, what the investigators did was they trained AI to learn the language of DNA. So they fed it different organism genomes, et cetera. So the AI was able to predict biological systems. So it's no longer just learning or writing computer code. It's actually writing biologic systems.
Interesting. So these 16 viruses target bacteria, not humans. And I want to talk about that distinction and how significant could this technology be in the fight against antibiotic-resistant infections? Because that is a problem that has been very difficult to find a way to address.
Yes, it is, John. So essentially what this is mimicking what happens in nature. In nature, we have viruses that fight bacteria all the time. In fact, we're surrounded by trillions of them. And what we have now in hospitals, like where I work, is an increasing problem with multi-drug resistant bacteria.
The Nobel Prize in Chemistry went to 3 scientists, David Baker, John Jumper, and Demis Hassabis, for their groundbreaking work using artificial intelligence to advance biomedical and protein research. The AI model they developed, called AlphaFold, uses databases with hundreds of thousands of millions of protein sequences to predict and even design protein structures, speeding up a months or years-long process to mere hours or even minutes. AlphaFold was rolled out just 4 years ago and has since been cited in scientific studies more than 20,000 times. Joining us now,
This is the world's first commercial biological computer, running on living human cells. The CL1 computer is the brainchild of Melbourne-based Cortical Labs.
This is where the neurons would be embedded in.
CEO and founder Han-Wang Chong says it fuses cell-derived neurons with silicon, creating a new class of AI known as synthetic biological intelligence.
Unlike using artificial neural networks, we grow real biological neurons into networks onto computer chips.
It sounds like science fiction, but Han says the CL1 is capable of learning and adapting faster than standard silicon-based AI, while consuming significantly less energy.
We take blood or skin, and we can transform them into stem cells, and from stem cells into brain cells or neurons that we then use them for compute and intelligence.
Han says the neurons grow on silicon chip with tiny electrical contacts connecting them to the digital hardware. And it's equipped with an artificial life support system to keep the cells healthy.
We have pumps like the heart, waste feeding reservoirs, filtration units like the kidneys, and we have a gas mixer.
Right, so you may be wondering why is a synthetic biology company spending its evening teaching this instead of quietly getting on with its own research. Because of a gap we kept running into, everything I have just described is published, open and free. None of it is behind a paywall or an export restriction, and yet almost none of it is being taught in Sri Lanka, and almost none of it is being used in Sri Lankan labs. The barrier is not access, and it is certainly not intelligence. It is that nobody has sat down beside our researchers and showed them how. That is a solvable problem. It happens to be exactly the kind of problem an education platform is for. And there is a deadline on it. Even though nobody has written it down anywhere, the global research economy is quietly repricing what a biologist is worth. The graduate who can take an open model, run it on their own data, and defend the result is going to be hired, funded, and published ahead the graduate who cannot. And the second graduate may well be better, may be well as a better bench scientist. That is obviously not fair. It is simply what is happening. As the first synthetic biology company in this country, we would be doing our job badly if we build the laboratories and left the people behind. Which brings me to our first speaker. It's none other than Dr. Lalinka Hera is the Chief Executive Officer and Chief Scientific Officer of Bioloom Islands, and he is the scientist who decided this country needed a synthetic biology company and then went and built a one. His training is genuinely international, a BSc in molecular biology and biochemistry from the University of Colombo, an MSc in marine molecular genetics from Jeju National University in South Korea, and a PhD in genetic engineering, completed as a split-side program between the University of Colombo and Deakin University in Australia with a research mobility scholarship at the University of Granada in Spain along the way. He is currently a postdoctoral research fellow at the Institute of Biochemistry, Molecular Biology, and Biotechnology at the University of Colombo. Alongside Bioloom, he is a visiting lecturer at the University of Colombo, University of Jayawardenepura, and ANC Education, and also a researcher at the Center for Advanced Material and Devices Department of Chemistry, University of Colombo. He has previously lectured at NSBM and at Wimber University. His own research run on genetic engineering and molecular biology. He is going to speak on AI biology, what these tools are actually doing inside working research program, what they do well, and where they still need a scientist standing over them. Dr. Lalinka, the floor is yours. Please go ahead.
Okay. Thank you very much, Udani, and very good evening, everybody. Hope I'm audible. Can you guys hear me well?
Yes, sir, we can hear you.
Okay, perfect. Right. So, right, as we came across about some areas as mentioned by Udani, and also you just glance in the videos we shared, the certain new technologies being mobilized, which are integrated with, you call that the artificial intelligence is being integrated in biological sciences. So we at Bioloom Islands decided that we should have a session, conduct a session on how these emerging technologies or the emerging tools are going to be integrated in biology, and also then to shape the nature of the biological ecosystems, how the biologists, young researchers, even the other scientists in the field, how they are going to position themselves in emerging era of these technologies. So I know a lot of participants in today's meeting who are the fresh graduates based in Sri Lanka, and also the still undergraduates who are curious about these technologies and excited to learn what are the trends. And we also named this pre-webinar session as the learn the language of biological future. So we will see why we named it so, and then go through the certain tools that we are going to discuss and understand learning or becoming experts in these tools, how it could support you guys in order to become or outdo the others working in the field and become the future scientists in biology, guys. So that's the idea behind the today's session. So we are going to develop our talk, our discussion, our conversation around 3 to 4 important tools recently released by certain IT giants that are useful in biology. We are all biologists, I guess, who are joining today. And we want to know how we are going to utilize or incorporate this knowledge, the AI-based tools developed by certain companies, how we could utilize them in order to understand how biological systems behave, how we can come up with new commercial products utilizing these technologies. Yes, as I mentioned, what I'm going to do today is not that I'm just going to introduce you guys, tell you guys about what tools are available. And I'm not going to teach anything in today's session as well, but to go down to the fundamental level and understand we ourselves, as biologists, how we are going to redefine ourselves in this emerging ecosystem of biologists as scientists. Back in the days, we know the biologists are supposed to be in the laboratories, go in the laboratories and conduct experiments, generate results, and based on your results, you want to interpret, you want to look at the results and understand something based on your observation. But throughout the development of these technologies, the way we look at the biological systems, way we conduct our experiments, way we understand how things work, those are becoming shifted to a certain different aspect, everybody. So that knowledge, if we do not understand these dimensions properly, later we will become left behind, as we mentioned, as biologists who are practicing outdated knowledge. The main idea is to understand how these new AI tools in biology are going to redefine the way we conduct biological experiments, and then understand what sort of new opportunities are being emerging in this particular biosciences areas. Right. To begin with, I would put this in this way that I might say that biology has always been a language. So, which means, so it's a logical, there are logical patterns in biological systems. In languages, we are trying to contrive sentences and try to communicate something, right? So in order to communicate, we need languages in our human language. The biology, when you look at biology as well, so you want to understand biological systems are all about information. And it's been communicated via a certain language, which is not human language. There are information written in biological systems. And then those are being transcribed, translated, and transmitted. So we have to understand the biology has always been acting. That's like a language. Sometimes I wonder why we are not trying to apply the certain rules comes in explained in linguistic in order to explain biology. Sometimes I think why these topics are not being taught at art faculties as well, because so it's a very creative area, the biology. To understand properly, we want to look at it in a different way, that you want to understand that it's about information and certain things are being communicated in a language. Your basic core concepts in biology, molecular biology, that you learn about DNA, where in a certain sequence of chemical, it's basically a macromolecule and a chemical where some information is being stored about biological systems. And you know there's another molecule called RNA. So that helps in, acts like a messenger and helps in spreading this information across the cells and within the cell and then certain areas in the cell. So it comes down to transcribing, storing information, transmitting information in a very interesting language. And of course, you go look at the protein. You know, those are comprised. The proteins are formed based on the amino acids, how amino acids are linked in a certain order. And it tells some information about how should that protein be looking like. So that's again, there's another language on protein. So if we could understand this language on DNA, RNA, protein, which means we can understand what sort of information is being formulating within that particular molecule, right? So that's what biologists have been doing so far. We are looking at this DNA sequence where specifically, precisely the molecular biology. They are looking at DNA, RNA, protein, what are the interactions between these molecules. And we are trying to understand this. We can read them, we can measure them, we can analyze what happens. So the first thing that we need to understand that it's a language we know it to learn in order to understand what's being communicated in a biological system. Now this fact was understood by the computational biologist as well. So, so if I compare what happened in past and what's been happening right now, back in the days as biologists, scientists, we ask a few conventional questions about biological systems. Like what structures does this protein have? What sort of structure a particular protein have? Right? So you ask that question in your basic bioinformatics classes. You want to follow the structure of a given amino acid sequence. Which sequence belong to this family? So that you can try phylogenetic trees and see which sequence are being aligned and what clusters are being formed. So you want to get an idea about how they look like, how they gather, right? What mutation might change function. So that comes in your cancer biology classes, in your disease-related, the genetic disease-related topics. You look at the mutation and you try to correlate. Once a particular mutation occurs on DNA, what sort of changes happens on protein, and then how would it direct to a certain disease condition? So we ask those conventional questions. But with the development of these computational tools that can be integrated in biology, can be utilized on biological problems. So we could come up with a few new questions to be asked. Can a computational model predict the structure without letting it know once the amino acid sequence given, without letting it know how the related proteins look like? Can it directly predict the structure of that particular protein, how it's going to be folding? What might the sequence encode? You asked previously which sequence belong to which family, such conventional question. Now we can also ask what might this sequence encode? A bit of intelligent questions, guys. Can we computationally explore consequences? Once the sequences change, what would happen? What sort of possibilities are there in the downstream changes? So we can now ask such questions because we have these intelligent tools developed, which means basically you talk about your AI systems, your generative AI tools like the basic ChatGPT, Claude, and the other AI tools. What you basically do is you use them in asking questions like a language model. They can just provide you answers via those tools. But what would happen if we could train a new artificial or new intelligent model to behave like a biologist? Now we have a biologist which is a synthesized new version, new biologist, which is not natural, that is written, that is trained. and it is an artificial tool. The students who discuss synthetic biology with us, they know in designing biological systems or in running experiments in biology, we used to practice as a core tool, which is the DBTS cycle in genetic engineering. In that, basically what we do is that we have to conduct experiments. And then based on the outcomes of those experiments, we get data, and then we can analyze those data and see what sort of scenario is there. And you can repeat the experiment. You can reproduce the same scenario, construct a new experiment in order to understand a certain aspect about biological system. Now, with the development of these AI tools, the DBTS cycle has been upgraded or modified, which is known as AI-driven DBTS cycle. Can you see how it's been modified? Now look at in computational biology, we can develop certain models, biological models, neural networks can be developed, and you can train it as a biologist, train on certain amount of knowledge about biological system and let it behave as a biologist who could predict certain aspect about biological mechanisms, metabolic processes, and how proteins folds. So the prediction component has been embedded into the DBTF cycle. And because we can predict, we can design things. We can now design AI-driven biological component. AI-powered tool can be used in designing biological segments computation. Once the design is done, you can move on to the experimental phase. It's been now expanded, the DBTS cycle in biology. We have a prediction phase and also we have a design phase. Our designs can also be tested in our experiment. Right. Because I mentioned that the biology is a language, guys. It's a language to learn. Biological system communicate a lot of information for us to understand as scientists or the biologists. Our job is to understand what biological systems say to us, and then even about diseases, even about physiological conditions. All these things are being communicated as a language. So this language, if it's a language, language has a grammar. And language could be in a different form. The biology as a language, so it can tell you a lot of information. Now, the foundation models that we specifically call these computational models we develop in understanding biological systems. These are foundational models that you can train them on different aspects about biologically sound scenarios. So we can basically train these models on big data sets, lot of data about biological mechanisms. It could be the DNA sequences, genomic sequences. We can maybe train a model, a neural network model on millions of genomic sequences in certain species. So the entire domain of life sequences can be fed into a model for that particular models to understand what are the information, grammar, what are the information written in that particular DNA sequence, and to intelligently look at the DNA. And if it's 14, we can train a certain computational model to understand how actually a protein amino acid sequence as the primary structure, how actually it could fall into a three-dimensional structure. Are there any patterns to be followed in folding that? Of course, as using human intelligence, we have a certain limitation. At a given time, we can look at only a single amino acid sequence and understand how that amino acid sequence could be folding into a certain three-dimensional structure. But these computational models that we train, they are powerful enough to parallelly process and parallelly understand how thousands, millions of proteins could fold into many different patterns, different three-dimensional structures, and are there any patterns they follow? And process those patterns, and based on those patterns they learn, they can create predictions about entirely new amino acids that we haven't seen before. In folding experiment, in our wet lab, we haven't studied that. But these tools are now capable of predicting how these new sequences can be folded because they are intelligent. They can make prediction, right? To learn patterns that enable prediction. If they can predict how a given sequence can be folded, why they can't generate entirely new sequences that do not exist in the nature? If these tools, the artificially intelligent tools are intelligent enough to predict something, if we could further improve them, why they can't generate entirely new proteins? Why they can't write entirely new DNA sequences? So these are the certain power, certain capabilities being incorporated into these training models. Right now, the computational scientists who are involved in biological science disciplines, they want to train these tools in a way that they can actually understand all these patterns in order to predict, in order to generate, in order to even design. Synthetic biology is our pet area. The certain the all our team attached to Bioloom Islands, they are working on synthetic biology, and they know our goal is to design new DNA sequences, gene constructs, and use them in environmental, agricultural, health applications. And we have to put a lot of effort in making sure that our designs are accurate. But what if we have the AI tools, artificially intelligent tools, which have already been trained as synthetic biologists, and they can come up with their own design? So you have your AI assistant working alongside you, designing new gene circuits. And as I show you, the validated design. So this is very interesting, guys. So how we as scientists, in order to deal and in order to understand these dimensions of the AI tools being emerge into the biological science areas, and what our role would be as biologists, as scientists in utilizing these tools. So that's the idea, guys, that these tools are being trained on these kind of aspects. If you compare, this is basically to understand that because this is a language, we can teach this language to so-called AI tools. Then they become experts in biology. Human language is basically comprised of letters, words, sentences, meanings. Likewise, we have analogy in biology. Biological language comprised of nucleotides, sequences, and you have folding proteins. Those are connected to certain functions. So this language, the ChatGPT and other tools are being trained on human language. What if you could train an entirely new set of models, tools on the language of biological science, the nucleotide, how nucleotide connect in order to store information about a gene, the promoter regions, the coding segments, terminators. This language could be learned by a new tool. So we could train them, right? Now I mentioned if they learn this, they can predict things. So this, I'm going to, as we mentioned, develop this story, develop what happens in the AI, I would say AI biology intersection. AI being slowly leaked, integrated, penetrated into the biological science discipline. So, three main ideas to predict information on DNA proteins, to design entirely new DNA sequences, right? And to build new DNA sequences. These tools can be utilized. So one of the main tools recently released, and we are very interested in this particular tool known as AlphaFold. And there's a lot of news on this tool as well, that even you saw that the inventors of this tool, they are from computational science background who became interested in understanding how a given amino acid sequence can be folded into a three-dimensional protein. So they were working years and years on this particular biological question, and they came up with a tool called AlphaFold, which could predict, without knowing the other information, it could predict precisely the accurate structures of any protein given, any amino acid sequences given into that particular tool. So this tool received Nobel Prize in 2024 because it was considered as one of the breakthroughs in science. That was the first time a Nobel Prize dedicated to pure science discipline, which is chemistry, was awarded to a computational tool. So the prediction done by AlphaFolders, that accurate, they had to give even Nobel Prize to this particular tool. If you guys explore the capabilities of AlphaFold tool as an AI tool, which can predict how protein could be folding into certain patterns. So as it explains, like for 15 years, it was a big problem for the conventional biologists. We had to go through this cumbersome process. You know, there are X-ray crystallographers working in the laboratories, and they use sophisticated platforms called NMR and cryo-EM techniques in order to determine the actual structure of an amino acid sequence, protein sequence. You go in the PDB database, you know that what being stored in PDB databases are X-ray crystallography data and also cryo-EM data, NMR data. So these are all very time-consuming techniques, and you have to need a lot of expertise and a lot of labors process to be going through in order to come up with a structure of a single protein. So you learn these techniques in your classes. But now the conceptual shift in here is that AlphaFold like tools are intelligent enough to computationally predict. When you give AlphaFold an amino acid sequence, because it knows the patterns, it's an expert in folding patterns, it can let you know how this given amino acid sequence could be folded into certain three-dimensional structure. So it can actually save a lot of money and time in science, guys, because these predictions are becoming very much accurate now. So we don't need to spend the certain money, time, expertise, and also labor on utilizing laboratory-based techniques in order to understand how a protein be folded. So this is very interesting and very exciting. Why exciting? I would say, now let's say you give a random amino acid sequence which does not exist in the nature. Because your tool knows how amino acid could be folding, it would let you know what sort of three-dimensional protein could arise out of that entirely new or certain synthetic amino acids. So do you understand these tools could behave like pretty much it's playing a God level information could be retrieved out of, I mean, they basically work and play God if you know how to carefully utilize these tools, the regulatory aspects and the ethical aspects. I don't want to cover in this particular session, as we want to give you guys an idea about how powerful these tools are and why it's important for us to learn how the techniques in AI biology and how to use these tools in order to become biologists in future laboratories. Right? Before the sequence, experiment structure, and now sequence, you can model, computationally model, and then understand the predicted structures, right? And then you can move on to the experimental stages, right? Another important question, what if AI could read the genome like language? I talk about with AlphaFold tool, an intelligent tool that could tell you how amino acid sequence can be folding into a three-dimensional protein structure. And talk about tools trained on the entire genomes. Genomes are very large and it processes a lot of information. Our genome contains all about our characteristic, our eye color, our height, the way we behave, our physiological processes, our metabolic processes. What if we could train a tool to understand and read entire information on the genome, written on the genome? The company NVIDIA, which is a giant in IT, they developed a platform foundational model called Evo 2. Evo type models are being trained in order to understand information on genome. It's being trained across all domain of life, which means trillions of genomes. It's intelligent enough to understand how trillions of bases patterns could be occurred across different DNA sequences, which means basically it's a species, it's a scientist, artificially intelligent scientist who knows in and out about how DNA can, any organism's DNA can store information towards certain biological process. How interesting is that, guys? So this is why we happen to come across certain news. The certain AI tool has written entirely new viruses which did not exist in the nature. Do you understand how powerful these tools are? And if we know how to properly use these tools, how we can utilize them in certain biologically important, something that helps humanity, right? Not to unethical aspect, but to find cures for diseases and so many other agricultural, environmental issues can be solved by properly utilizing these trained tools. Now, first I told you about AlphaFold, which is trained on protein folding, understand how protein folds. And now this tool I let you know about is NVIDIA's Evo type models. These tools are their intelligent genomic platform, AI tools which are genomic scientists. Now you can work alongside with them. You can use these artificial intelligence scientists in order to find answers in your research questions, in order to design something new, right? So this is the interesting aspect. So these are the areas we should be exploring in near future in order to excel in biological science, of course, the conventional laboratory techniques are very important, right? And another platform I need to pay your attention, grab your attention on, you have tools intelligent enough to understand how proteins being fold. And also we have tools as molecular biologists, we have tools now to understand how once an entire genome is fed into that tool, it knows every single biological process encoded in that particular genome sequence. So very easy to work with that. You ask the question with that tool, then it will let you know these kind of physiological processes are being scattered across that particular genome that you don't want to go gene by gene manually in order to understand with the conventional tools to analyze and get an idea about certain biological aspects. So if you work with Evo tool like model, now think about the AI co-pilot. What you call co-pilot? So there are different tools, expertise in different aspects of biology. Let's say there's a platform that can incorporate all these scientists. This is more like contriving a group of scientists that work together, that to develop your own research group into a single platform. You have a protein biologist, you have the genomic scientist, you have another bioinformatician, and you integrate all of them into a single platform and let the process run as a pipeline. So this been done recently by the certain, the Anthropic is a very popular AI giant, right? So Anthropic as a group, they decided to incorporate everything together and come up with an AI scientific co-pilot called Claude Science. So this is a very powerful platform that integrates everything into a single platform. Entire drug discovery pipeline has been integrated into a single platform. You give the target, you get the drug from the other corner. So how, if you learn the computer-based drug design or a conventional drug design process, in order to validate a single drug towards a certain disease or a disorder, it takes 10 to 15 years, guys. So that pipeline has been, the timeline has been significantly shrunken due to the ability of these kind of tools to integrate all these components into a computational pipeline and get the output out via a very intelligent process. So that's why we say, so these platforms now exist alongside us. So we as biologists, we learn biology in the classes, in our university now, maybe in certain programs. So what to learn? We have to rethink what we have been learning. Are we trying to compete with these tools, or are we trying to learn how to utilize and how to collaborate with these tools and become some expert in future science? So that's the idea, because these tools are being emerging. So as biologists, we should understand how we are going to incorporate these in our workflows in the laboratory. So the compute is moving into the laboratory, you see. So back in the days, the biological science laboratory is all about wet lab tools, that you have certain incubators, you have certain, the incubators, the PCR machine, of course, in the recent laboratories, but it's all about wet lab techniques. But have you seen nowadays that you get a small computer science laboratory as well, the bioinformatics corner in your laboratory? Do you understand what we've been doing as wet lab scientists was what physically simulating these biological processes, but as computational scientists, we are starting to simulate the exact same thing based on the experimental data at the bioinformatics phase. I'm not talking about AI mediated phase. before that, we use computational platform in order to simulate the sex. Exactly the same as me. Now you are moving into an era where they do not need human input anymore. They are intelligent enough to understand what's to be processed. The entire simulation is being done in the platform. So the border, the margin between the certain border between wet lab and dry lab has been dissolved now. So these, I would say, we used to call wet lab scientists, dry lab scientists, but I would say now the scientists in biology should be called as what? So I coined a new term. There's a certain phase, right, in between solid and the wet phases, liquid, the plasma state. We would come up with a term called plasma lab scientists, because we are integrating, we are the bridge between wet lab aspect as well as the dry lab aspect, and we know how to intelligently connect these two aspects and come up with something significant. So we want to fundamentally understand this shift from certain wet lab to dry lab to, after that, the intelligent lab in dry labs, right? So if we do not carefully process this aspect, we would not be able to survive everybody. In sooner or later, the scientists who do not practice these emerging tools, they would be slowly shifted out from the system. So this is why we thought of stressing the importance of learning in real-time manner, the tools being emerging and being discussed in global scientific ecosystem. Right. So let's see today's scientists, what have they been doing? So, so this is very familiar. This list of tasks I have under today's scientists. Search literature manually, you go to now the AI-powered literature mining tools are there. I know our young researchers they of course use these tools. That's what need to be happen, guys. Search literature manually, then write code from scratch. We were writing, even the computational IT people, they used to write codes from scratch. So it was like Greek for biologists. Now we have ways of importing entire codes and get some biological processes done, thanks to these AI tools being trained on the biological science aspects as well. Design one experiment at a time. So that's what we've been doing. Wait weeks for your results. And iterative cycle is very slow. Think about emerging modules that we've been discussing here. So it would define biological problems precisely. The Claude science will lead to understand very philosophically sound questions, how to phrase-wise elaborate such questions if you start working with these platforms, right? The integrates of foundational modules that we discussed, Evo 2, AlphaFold, and you can explore vast design space in synthetic biology related topics. The design could be powered by AI tools, right? We were in the discussion of developing, training new design tools. So our students who are interested in these areas, of course, they focus on developing AI-powered foundational models that they could design entirely new biological systems and simulate how it would happen in real world. That's what we do with simulations under real biological conditions. So we can also simulate with these tools, and we validate and feed results back into the iterative cycle. So we have to change our mindset and see how we are going to redefine our design build cycle even in biological commercially relevant biological processes, as well as when it comes to pure science as well. How we design our pure science experiments with the support of these AI platforms. Right. So that's the core idea, everybody. We want to understand if we do not learn these tools today, we would be left out in the Bible. I recently came across that there certain international research groups, they announced their new roles as intelligent CAR T cell designs, which means CAR T cell therapies should be designed using AI tools. So we don't even, some of our students don't even understand these job roles are being announced for biomedical science, biotechnology students, because they announcing in a different way that we are very much behind. So we don't want that to happen in our ecosystem. So that's why we decided to spare time on exploring these tools with our young researchers and also students who are involved in these areas. Of course, while practicing these conventional techniques alongside simultaneously, we want to spare time on learning these platforms as well. That I would say in order for us to survive in future science. Right. The language is changing, the biological language was something that we read manually before. Now it's being changing and opted into AI-powered reading platforms. So likewise, so these three models and one platform. We also have very interesting, exciting other platforms we will also be exploring in our upcoming sessions. But here we are going to focus on AlphaFold, NVIDIA Evo 2, Claude science platform as a co-pilot in science, and Cortical Labs CL1. So that's a very interesting approach that Cortical Labs have been developed. They want to redefine the entire logical platform of AI-powered tool. It's been trained on neural network before artificial. Now, Cortical Labs want to use actual human neurons in order to develop neural networks that can be used as biological computers. And they can apply the theories in neuroscience, neuroplasticity, neuroregeneration theories in computational biology in order to develop evolving platforms of biology. So we want to explore all these platforms and to understand how future biological ecosystem would be shaped and what would our role be as scientists. Right. So that's about the introduction to these tools, guys. Hope we had got some idea about what we are trying to discuss throughout this process. So thank you very much, everybody. So the discussion is open to you all, and also Sanjula has a session after me on the technical aspects on these tools and what are certain areas, because they are using these tools in their research route now. Okay, thank you very much. Over to you, Dani, Sanjula, and team, everybody. Bye.
Yes, so Dr. Lalinka, thank you, and that was an excellent session. And I think the part that will stay with the people is I can see that the chat is filling up already, which is exactly what we wanted. Please keep those questions coming. Dr. Lalinka will be back with us for the Q&A session, so nothing you post is going to waste. What Dr. Lalinka has just given you is the why. The obvious next question is the how. What do you actually sit down and do on a Saturday evening in September to get from where you are today to using these tools on your own work? For that, let me bring in the person who built the workshop, Mr. Sanjula Aryaratna. He's a lead scientist at Bioloom Islands and PhD candidate in synthetic biology at Wayamba University of Sri Lanka. He is here on behalf of the Bioloom team to take you through the workshop itself, and he will be the one of the people running the sessions on the 5th as well. So if you have a question about what you will actually be doing on the day, he's the person to ask. Sanjula, over to you. Take us through it.
Okay, thank you, Dani. Yes, good evening, everyone. Yes, so Dr. Lalinka just given you the why we are doing this, like why we are doing this session, and my job for the next few minutes is and how we are going to do the workshop. So let me share my slides. Yes, I hope you can see my screen well. Yes. So yes, I'm not going to read you a list of features about these tools, right? So that can be obtained from any website, any news platforms that we showed you, like in YouTube, there are tutorials and everything is there, right? So I'm not going to like read you that list. So what I want instead is that when you leave tonight, you understand what each of these tools is actually doing underneath. Okay, so because that is the difference between someone who has heard of, let's say, AlphaFold, someone who had heard of AlphaFold, and some who could sit down and use it at the workshop. Yeah. So on the 5th of September, we will work through one biological engineering problem, okay, from beginning to end, using these 4 computational platforms in sequence: Claude Science, Evo 2, AlphaFold, and Cortical Cloud. We are going to carry out one case study throughout the whole way. Okay, so and the case is like the case study is engineering an improved fluorescence reporter for field deployable biosensor. Okay, let me put the problem in front of you properly. Okay, here is the situation, guys. So let's say we have a fluorescence reporter protein and it is being used as a part of a biosensor. So let me let you know about what is a reporter protein. So reporter protein does something like conceptually simple. Okay, so it converts a biological event into a signal that we can detect. Let's say something happens, the reporter fluoresces, as in the picture, reporter get fluoresce and we can read the fluorescence. Okay, so in a control laboratory that can work, but our case study is we need to like in a control laboratory there the temperature is stable and the buffer is what you have decided would be, right? So the sample is also clean. But if we are going to include this reporter protein in a biosensor, that is actually useful to work somewhere else, okay, in the field, or let's say we are using it for like coastal aquaculture biosensor, or else we are at a rural sample point. So the conditions that nobody controls, right? Those conditions we cannot control, like in a laboratory. So under these conditions, the protein has to hold up. So the question we'll spend on the workshop is like, can we identify the protein variants that could potentially provide the better structural stability or more reliable reporter behavior outside the lab? Okay, that is the case study that we'll do using these tools. So now also I mentioned the important word in here is potentially, and we want to be precise about it. Okay, so because it defines what kind of workshop this is. Okay, so on the fifth, at no point we will claim that an AI prediction has improved this protein. We will not have made a protein. We will not have expressed one. Okay, we will not have measured anything. What we'll do is we use computational evidence to decide which variant are scientifically interesting. Okay, so, and I will say plainly, this is not an AI tool demonstration, guys. We are not going to put a sequence, let's say we are not going to put a sequence and AI into an AI and accept whatever comes out. Okay, what will we will treat these systems as instruments. Let's say you have instruments, right? In a laboratory, you have various instruments. PCR machine is there, in a molecular laboratory, PCR machine is there, spectrophotometer is there. There are many other tools, right? We are going to treat these tools as instrument as well. Okay, so yes. Throughout the whole workshop, we will keep 4 questions running. What is the biological problem? What does the model actually know or actually predict? What can we infer from that prediction? And what would we still need to experimentally validate, guys? So the fourth question is the discipline. Okay, so if you can answer it at any point of the workflow, you are doing it correctly. Okay, if you ever if you find yourself thinking there is nothing left to validate, that is the signal that you have correctly started treating a prediction as a result. Okay, so that's distinction between this prediction and an experimental validation is the single most important idea in this whole workshop. Okay, so the first tool is Claude Science. Okay, the easiest mistake that you all guys make treating Claude Science as another chatbot like ChatGPT and other tools like Gemini. There are also in Gemini, there are many other things that we can do as well, and ChatGPT as well. But we should not treat Claude Science as another AI tool, AI chatbot. Okay, so that is not how you will use it in the workshop, guys. So for this workshop, Claude Science is a scientific reasoning and evidence synthesis layer. Okay, it is the thing that sits between a vague biological wish and a structured engineering problem. Okay, so and let me show you why this layer need at all. Okay, let's say so I want to improve this fluorescence reporter. So yes, so I'm going to type in the Claude Science. I want to improve this fluorescence reporter. That is biologically interesting, right? People say it in lab meetings constantly, but computationally, it is not yet a problem, right? Because it does not say what to optimize or how you would know that you had succeeded, right? So you just basically saying, I want to improve this fluorescence reporter, but we do not know how to improve this one, right? So let's say you want to improve it in a way, let's say a brighter signal per molecule, a high fluorescence, better folding. So you can make it more functional, right? So high thermal stability and let's say low background noise, better expression yield, and also it is resistant to degradation. So those are the qualities that we expect from this fluorescent reporter if we put it in a biosensor, right? So like that, and we are putting several of these together to get a more stable protein. Okay, so yes, but there is a rule. So we are using Claude Science for this, but we have a rule. So I want to hear it tonight rather than on the day of the workshop. Okay, so because it's the thing that make this as a scientific workshop rather than a software tutorial, right? So let's say your ChatGPT or your Claude Science says there is a mutation. Okay, so we are like looking for reporter variance. No? So let's say X kind of mutation should improve the stability. So they are giving it, right? They are giving it in the chat. But we will not accept that. We will interrogate it. So what are the evidence that support this? What is there any literature showing this one? Okay, so is there, or is it an assumption? So is the residue conserved across the homologous? Because like conservation itself is an evidence, right? So that's how you should like go through the Claude Science. So we will, like in the workshop, we will give you the whole tutorial to yourself, and you can follow us with these different steps. Yes. And this brings me to what I think the most transferable thing in the whole workshop. So we will separate three things that normally get like combined together. Okay, so one thing is evidence. So evidence is, yeah, so evidence is something supported by a source or database or an established observation, right? The literature shows this residual participate in an important interaction or inference, a conclusion we can reasonably derive from that evidence. Speculation, something that could be true, okay, something that could be true but not have been established, let's say. So there is no like measurement behind it. For example, this fluorescence mutation will increase fluorescence by 30%. Okay, nobody knows that, right? There is no measurement behind it. That's the thing. So we are speculating that. So, and here is the difficulty. Yeah, right. That is the difficulty behind this one. We will let you know how to solve these problems in the workshop with Claude Science platform. Okay. So then, so you will leave this section in the workshop. At the workshop, you will leave this section by end of this first tool session. It's like it will give you your biological problem stated properly, the properties that you want, and the properties that we should document, and candidate mutations. Then we can go into the next tool, NVIDIA Evo tool. So we did the scientific reasoning, and now we are into biological sequence modeling, guys. So we are modeling this sequence. So to understand how this Evo tool actually works, okay, how this will work. So you need the problem it exists to solve. The problem is sequence based, guys. A DNA sequence is not just a string of letters. It represents the whole biological information, as Dr. Lalinka mentioned, right? Through a chain we all know, but rarely state carefully, okay? So now the number of possible sequences are enormous. let's say for a protein, the length is equal 20. So we are like testing every variant here. Okay, so in here we are going to test the whole variance, like we are going to compare the whole variance that we got from Claude Science. Let's say we can take a protein about 230 to 200 to 300 residues. So that is the roughly, we can say it is a that is the rough length of a fluorescence protein. Okay, so so the sequence space of a one protein is like this is really large, guys. So like let's say this fluorescence protein is having 200 residues. So 20 into the power 200 is a number with over 300 digits, right? So there are something like 10 or 18 atoms in the observable universe. So this sequence space of one small protein is unimaginably larger, which means the exhaustive approach that we are going through is not funding problem. It is physically impossible. So what does this Evo 2 gives us? A computational way to explore that space. Instead of experimentally generating thousands of millions of variants, we computationally examine the candidate sequence changes and which one looks more plausible according to the model. And it is worth knowing what that model actually is. So Evo 2 is a foundation model trained on organism genomes, right? So in order of here, we can hold the relationship between the genes and the regulatory elements away on the same chromosomes, and it is fully open. Okay, so it has learned essentially what is a real genome is, guys. So which sequence has sequence evolutions has tolerated, which it has not. So when the model gives a sequence, a high likelihood the honest reading is yes, this is something can exist, right? That is something can be exist. Okay, so before generating any variance, okay, before generating any variance, you need to know about what is a mutation. Okay, so how it will be useful. So you need this molecular biology aspect to go through it. So take this codon like GCT, and if we change the third base, GCT becomes GCC. But these codons code for alanine, whether is it GCT or GCC. But let's say we are changing the second position, so then alanine become valine. Okay, so these things you need to go through, and we will also give some idea about this one as well. And yeah, so what we are going to actually do with Evo 2 is we are going to start from the reference coding sequence that we obtained, and we are going to explore candidate variants across the sequence, and we are going to examine the differences between those variants and our wild type. So we are going to narrow that enormous space that I mentioned, like we could have to study millions of variants into 5 to 6 candidates. Okay, that's what we are going to do with Evo 2. So the next tool is AlphaFold, guys. So we are going to now we are in an interesting position. we have a sequence. Now we have variance. So then we are going for the so in AlphaFold we are addressing we are predicting the structure, right? So AlphaFold addresses one of the greatest problems in structural biology. So like we are giving a protein's amino acid sequence, can we predict its three-dimensional structure? And remember, but this is an like prediction. We are not directly observing the molecule. Nobody has put like variance in a beam. What comes out is a structure model. Like on here, like in here, like we will examine the wild type and each variance, and we will like predict the structure of that protein. Okay, so like there are like two confidence matrices. Okay, these two confidence matrices. So like in here, the first metric is PLDTT. In there, like we are this is the predicted local distance different test. So it is reported per residue on a scale from 0 to 100. Okay, so it tells you how confident the model in its own structural prediction at the position. So the next one is PAE, right? Predicted aligned error. And this answers a genuinely different question. So like PLDTT is on how confident I'm about this residual now. But PAE is how confident am I about where two regions is relative to each other. It's pairwise, reported in angstroms, and you read it as a two-dimensional matrices rather than as a value along the chain. Okay, so we will go through mutation mapping as well. We will go through like DNA mutation, amino acid substitution, 3D location, and structural hypothesis as well. So like we will integrate all these tools, like all this data together, and we will ask these questions, guys. How plausible is this sequence? How does the sequence model suggest? And what does the structural prediction suggest? How confident is that structural prediction? And is there biological or literature evidence? What are the risks? So that's how we are going to conduct this case study using these tools. Okay, now it's about the cortical cloud. Okay, so everything we discussed so far has used computational models to understand or engineer biological models, right? Molecules. So with cortical cloud, the arrow is getting like changed. Okay, so we did computational modeling to understand the biological molecules. But using this cortical cloud, the arrow runs the other way. Can biological systems themselves participate in computations? So as you saw in the video, these cortical computers, we can actually physically buy these computers. Also, we can access them through their cortical cloud server is there. So we will use that in our workshop as well. So you will have the chance to access the cortical cloud and see how biological neurons can help solving the problems. Okay, so like as I mentioned, like in conceptually in here, like stimulus, let's say a stimulus into the biological system, we can get a response, right? We are acquiring their signals, and then we can get a computational interpretation. So we are going to use this Cortical Cloud to compare and like compare our evidences, and we will going to use this to our field biosensor. Okay, so and that is what we are going to do, and we are going to connect this whole Claude Science, Evo to AlphaFold, and we are going to integrate all those together and get the results as we want, and we are going to use Cortical Cloud, and that is the whole workflow that we are going to use in this workshop. Yes. So if you have any questions, you will like in the Q&A sessions, you can ask from me, and Dr. Larinko is here as well, also here as well. So yes, over to you, Dani. Thank you so much, guys. Good evening.
So thank you, Sanjula. So that is the whole picture starting from the 5th of September. You leave having actually run these tools yourself rather than having watched somebody else run them. So now, as we promised earlier, this is the part I ask you to stay for. The next gen workshop is priced at 5,000 rupees. For everybody who attended tonight, we are bringing that down to 3,500 rupees. So we'll be sharing the code on the screen now. I will leave this slide up while until we do the Q&A session, so it stays in front of you for the rest of the session. But please do not leave it until the very last minute. So then let us open it up to Dr. Lalinka back with me. I have been collecting your questions, and you can keep sending them in as well. So if you would rather ask live, raise your hand and I will unmute you. As the first question, I have selected this one. Dr. Lalinka, can you hear me?
Yeah, I'm here. Yeah.
So as the first question we have, do I need to be able to code to get value out of this workshop?
No, Danny, not really. That even I mentioned throughout my session as well. So to generate code, we have a lot of other options now. So this is for biologists, guys. You don't need to be scared about your level of coding or anything. Sanjula, do you want to add anything to that?
Yes, we don't need any coding for these sessions.
Free knowledge in coding, right? Yeah.
And we have I am a final year undergraduate, so I'm from an non-biology background. Will this suit for me?
Of course, the IT people who have been exploring how they can test out their knowledge in IT ability, of course, they can see how they can actually join the workshop and understand biological science concepts and to learn how these ideas that you are going to use your knowledge. Let's say you are IT student, you can of course learn these tools and see whether you can explore any opportunities in biological science field as well, right? So as an example, these tools being developed by IT science, IT students like IT companies and NVIDIA, Google, and also so these are all such teams work together. These are interdisciplinary areas. So of course, even engineering students, they can join. Medicine students, they can explore these topics. Yes, Udani, they're from other areas. They can definitely join the workshop and learn the tools that we are going to teaching the session. Right.
Yeah, from that, there's another question. Can these tools run on an ordinary laptop or do I need a GPU?
It depends. So certain tools can be run. Sanjula, do you want to specifically mention what sort of computational power we need?
Yes, sir. Actually, we do not need we were going to run it through server based one, so they can use their ordinary laptops for this one as well. For Cortical Cloud, we will give access, and for the Cloud Science, we will give access, and the SDK files will be shared through an email for the registrars participants.
Yeah, so there's another question as are the tools you showed genuinely free or free until the point where they become useful?
Sanjula, again, over to you.
Yes, actually, the NVIDIA Evo 2 is not a free tool. We requires uh, like some $20, $30 like points to use that one. But for the session, we will have it the participants will have the access for the NVIDIA Evo 2 as well. Others are like Cloud Science is free, and Cortical Cloud we will give access, and AlphaFold is a free tool that we can use. Yes, the participants will get the exclusive access to the NVIDIA and Cortical Cloud platforms.
So the next question is regarding the AI predictions. How do I know an AI prediction is right? What stops me publishing something wrong?
Yeah, to answer that question, Udani, so in biology, even we have to cross-validate with multiple tools. Even in the conventional bioinformatics aspect, what we do is that we basically run the same simulation with multiple tools and cross-validate. And of course, you can cross-refer with your experimental data as well, if anything available. Accuracy in the sense AlphaFold is currently recognized as one of the very much accurate tools in prediction aspect. So it passed certain tests in predicting the folding aspect of protein. So of course the data generated with these prediction tools, AlphaFold like tools are being, those are acceptable in certain, any kind of, across any kind of international journals. To search, you will come across that there are predictions done by, it depends on your actual application and insight you are trying to obtain, guys. So case to case, you might have to experimentally validate it, or let's say there's no sequence available in the any predicted experimental structures available in the database. So in that case, you should be able to justify why you use that prediction aspect as the only option in your study to the reviewers of the journal. It depends on such, it's conditional and depends on the situation. Right.
And someone asked that where does this leave wet lab skills? Is the bench becoming less important?
That is something we all can together discuss. Sanjula, what do you think about that? Does that mean bench becoming less important, or it reflects the same aspect as I wanted to mention? So it's very interesting that lab is lab, that it's not a kind of competition or a battle between the wet lab scientists and the computational scientists, but we are all, both these groups are trying to work towards a single goal, right? To answer biologically sound questions, right? So integrating the wet lab aspects with computational aspects. And I would say now we have become the plasma lab scientists, as I mentioned. I started as a heavy wet lab scientist. Now we are in computational biology. We practice all these tools, micro injection, next generation sequencing. At the same time, we are into these design tools, simulation tools. So we want everything, anything that supports in answering your question precisely in biology. That's the ultimate goal, right? So it's not about whether the wet lab is. It seems like wet lab is getting replaced. The wet lab is becoming automated as well. Sanjali, do you want to come up with a few examples on
Yes,
companies are doing this?
yes, like Ginkgo Bioworks, they are doing a pretty good
Jinko
job.
wants
Yeah.
to automate all these wet lab workflows. So Jinko, if
Yes.
you go to Jinko's website, you will see amazing things that they've been doing. Right? The
Yes, like
microbiology workflows and all kind of workflows are getting automated, and no longer we want to touch the pipettes with that input.
yes, like even like using the Claude Science tools, like we can connect them together. Like we can connect the AI tools with the instruments. Like as I know, like they can like now optimize the PCR workflows, assay development, everything they can do their own.
Even cloud science, they are aiming of replacing entire conventional drug discovery pipeline as well. So
Yeah.
that's the million billion dollar industry.
Yes, yes, we showed that one in the news. Like
Yeah.
that is the ultimate goal
That
of.
Anthropic CEO's ultimate goal. What's behind the screen happen.
Yeah.
All right. So, as the next question, I have selected this one. What can I realistically do with this in a Sri Lankan lab on a small budget?
Sanjula, you are working in Sri Lankan's lab with big budgets, though. Do you want to answer that question? Yeah.
Yeah, like even in my research as well, I'm using these computational tools, and we are going through several tools, like not only these Claude Science or AlphaFold or anything. We are going through many tools, like designer tools, simulation tools, before we are going for a wet lab part. Even we are going to publish, like we can publish those data in journals, like uh, yes, because they are now like really uh, accurate. Okay, so then we can go ahead with the like before the wet lab part. As I mentioned in my workflow, like when I'm telling about this workshop, so this is a prediction, guys. This is a prediction, so that we can make predictions to less the like we can do less work, wet lab work later by using these tools. That is the goal of behind these tools as well.
So the high throughput phase of
Yes.
analyzing can be narrowed down to one or two experiments at the same time. Alongside in designing, these are very important that you guys as synthetic biologists, you all are working on design and simulation.
Yeah.
Uh, uh, you all are developing gene circuits, right? Sanjul and even our other students for cancer research, agriculture, biosensors. So those simulations, of course, you can publish as entirely full-length research article, right? First, in good journals, guys. Then move on to developing it. So which means so that's very important. The initial computational component of developing and designing.
So, we would like to keep the Q&A session open for last few minutes. And we would rather hear from you than from us. If you are working on something where you think one of these tools might help, or if you tried something and did not work, please unmute and tell us. That is genuinely more useful to the people listening than anything we prepared. If you want to talk, please let us know. And In general, there's this question, will there be more workshops after this one?
Sanjula?
Yes, actually, after this next session, like this next gen workshops will be go on, but we will not hold any typical other workshops. But this next gen workshop series, as we know, as you guys know, like we held a session for POCD devices, and we went through eDNA and metabarcoding session at BMICH. So this next gen workshop series will be go on, and we will study the timely science, and we will study future aspects, the next generational aspects of biology and biotechnology, and we will go on with this that will go on with this next generational workshop series. So
We don't want to
anything?
go back to science, right? We want to explore what are emerging in the future.
Yeah.
Throughout our next gen workshop series.
So, like that, those were the most general questions we had. With that, we are at the time. So, let me close. Thank you first to Dr. Lalinka Hera. That was a great deal of substances with all this time. And thank you to everyone of you who gave us your evening. Biology has spent of its history as science of description. It is turning into a science of design, and that shift is happening inside our working lifetimes. There is no reason at all for Sri Lankans to be watching it from outside. We would much like to see you on the 5th. Thank you and have a good night.